Okay, hello everyone. Thank you so much for joining us today for the e-seminar series in Translational Biomedical Engineering with Professor Kibret Mequanint from Western University. He's going to talk about designing biomaterials for therapeutic goals from radiotherapy, dosimeters to tissue engineering and to bioadhesives. But before we go to his talk, just like always, we go through some housekeeping note during the talk. You can put your question in a Q&A box just on the bottom of the screen. There is a ask a question box, and there is a chat box to greet each other, and then you can introduce yourself, say hi to your other colleagues, and get kind of connected to each other for any collaboration or any kind of efforts that you have, scientific efforts you have. So next speaker is Professor Lindsay Fitzpatrick from Department of Chemical Engineering at Queen's University. She will talk about modeling tissue damage and toll-like receptor signaling in macrophage biomaterials interactions to get the most updated information of the upcoming e-seminar series. You can follow our Twitter account with the hashtag handle TransLBME. Or also on the LinkedIn, the co-organizer, myself, and Dr. Akbari, like we post the upcoming seminar series. So last but not least, I would like to thank also our sponsor, Montreal Transmedic Institute, which sponsored this seminar series from almost the beginning in June 2020 that we started this seminar series. So by that, I have the honor to introduce our guest speaker today. Professor Kibret Mequanint is a professor in the Department of Chemical and Biochemical Engineering and the School of Biomedical Engineering at Western University in London, Ontario, Canada. He joined Western in 2003 after completing his PhD from the University of Stellenbosch. I don't know if I'm pronouncing well, in Cape Town, South Africa, and two successful postdoctoral fellows from Darmstadt Technical University in Germany and McMaster University in Canada. His research interest is in the area of biomaterials, tissue engineering, regenerative medicine, bioadhesive, and non-diffusing radiotherapy gel dosimeters. He has authored and co-authored more than 130 peer-reviewed journal publications, over 70 conference proceedings, and several patents that have been licensed. His contributions to biomaterials and regenerative medicine research have been recognized by a number of prestigious awards, such as Early Researchers Award from Government of Ontario, Engineering Excellence in Research Award from Western University, Faculty Scholar Award from Western University, and Bikilia Research Excellence Award in 2014. In addition to research, Dr. Mequanint is a dedicated teacher and mentor, receiving both Western's Faculty of Engineering Highest Teaching Award in 2011 and Western's Edward Pleva Teaching Award in 2015, the highest teaching award at the university of level in the faculty. In terms of academy membership in 2018, Professor Mequanint was elected to the College of Fellows of the American Institute of Medical and Biological Engineering as one of the top 2% of medical and biological engineers. In 2021, he was elected to the Ethiopian Academy of Sciences, and in 2022, he was elected to the Canadian Academy of Engineering. Professor Mequanint is the active member in the Canadian Biomaterials community and served as the president of the Canadian Biomaterials Society. He is an associate editor of Frontiers Vascular Physiology and an editorial board member to several biomaterials-related journals. And this list goes on, and Professor Mequanint is our own colleague and friend in CBS community, and I have always been learning from his work and his kind of mentorship and leadership. Thank you so much for accepting our invitation. So you can share your screen with us and start your presentation.
Can you see my screen?
Perfect. Yes.
Okay. Well, thank you very much, Humon, for your very generous and kind introduction, and also for inviting me to give this e-seminar on biomedical translational research in biomedical engineering. I also want to thank you and Mohsen for your leadership to put this thing together. As I understand that this e-seminar has been going now for almost 2 years, and that's a wonderful thing, and I'm sure researchers here in Canada and elsewhere will benefit from this platform. So thank you again for your leadership. You can see that the title of my talk is fairly broad, and the reason for that is because I would like to point out some of the translational activities that my lab has been working on in the past. Just to give you a summary of what my lab is doing, the overall activities in my lab involve these two categories. The first category is in fundamental studies, and these fundamental studies can be, again, broadly classified into these three areas where we have biomaterial synthesis. Let me just pick up a pointer here. In the area of biomaterials preparation, we synthesize new and emerging materials, hydrogels, biostable, biodegradable materials, and hybrid materials, both organic and inorganic. We also are involved in the fabrication of scaffolds using different techniques, solvent casting, particulate bleaching, electrospinning, and other forms as well. And the most important thing then is to combine the biomaterials and the scaffolds with cells to do cellular engineering. And in this area, my lab has been working over the last almost 20 years now in the area of vascular and bone tissue engineering. So most of you probably know my lab through the work that we have done in vascular tissue engineering, which I will be talking very little about it today. Then, of course, once the fundamental studies are done, then we are interested in translating the fundamental studies into something that is useful. And in the area of translation, again, you'll see these three categories. The first one is radiotherapy quality assurance devices, which I'll be talking to you shortly. And then we have tissue bioadhesives, and then, of course, engineered vascular tissue models and injectable devices. Now, this is not a perfect equilibrium, as you can see from these double arrows in here. The shift could be, sometimes the fundamental studies are more important, and then therefore the focus sometimes is in the left side of the screen, and sometimes we get partners from industry where our focus is mainly on translation. So I don't want you to see this double arrow here as a perfect equilibrium. It's not. But generally, we go back and forth between these two categories. So what I am going to start this presentation is basically taking you to the beginning. And this is many, many years ago now. And that is me in this picture. And you have to trust me that it is actually me. This is during my early day in industry before I even go to grad school. This is a commercial flyer for a chemical trade show that was prepared, and this is in English, and this is in German, and then this is in Portuguese, because the company wanted to prepare this particular flyer in 3 languages, and so this was around mid 1990s, and you would see me basically playing around with 4 different reactors. There is one in here and just one behind me. And then there is the one that I'm working, and then of course you can see the 4th one in here. So I was involved in a polymerization reaction lab for the private sector. I briefly worked in the private sector before I go to grad school. And in those days, I was involved in polyester polycondensations, polyurethanes, polyphenols, polyacrylics, and silicon polymers. And all of these polymers were actually used for the metal finishing industry. And the company that I was working in the mid 1990s has disappeared because of rebranding and amalgamation with other companies. So the primary goal during those years was to develop polymers or generally polyesters and polyurethanes for the most part. They are dissolved in organic solvents, and that polymer is then used to apply as a very thin coating to protect metal surfaces from corrosion. So this is a very large industry. It's a multi-billion-dollar industry, and the industry is specifically called coil coating because the metal is rolled in the form of a coil, as you can see in this picture. So I was thinking about how I can eliminate some of the organic solvents that I was dealing with every day. And one of the questions that came to my mind was, can I actually use water as a solvent instead of using these toxic organic solvents? And so that basically led me to go to grad school, and that's basically the beginning of the story in terms of my translational research. So for those of you who may not know the process of metal finishing, it involves a heavy machinery. Sometimes you really need to take a short ride to go from the beginning of the line to the end of the line in here. So these heavy metal rolls are loaded using machinery to the platform in here, and they get unloaded, and then they go through a series of processes, one of which is just to uncoil it. And then there is an accumulation, 2 accumulators actually, one in here and one at the exit. And this is, basically if something happens in the process, then instead of turning everything off, you can just increase the level of these accumulators, and so you can still run the plant. Now the process involves a chemical modification step that is known as phosphating, which is widely used in the metal treatment industry for surface finishing of ferrous and non-ferrous metals. Now, you can see that this process in here is a pretreatment or a cleaning, sometimes called a conversion coating. The phosphate conversion coating results in a non-conductive layer of metal phosphate, in a crystalline form, and that crystalline metal phosphate is non-conductive, and then therefore it prevents the metal from corrosion. So that's really the idea. But this process is rapid at the beginning, but kinetically it becomes very, very slow towards the end. So it never really goes to completion. And so that was the challenge that we had. And in most commercial treatment lines, oxidizing agents and accelerators are often added, and so the waste is then disposed to the environment. So you would imagine that environmentally it was not a sustainable process. So the idea came to me that the best way to deal with the environmental safety is to get rid of this process. Now, this process also uses chromium as a final stage in the sealing process. And when chromium is disposed into the environment, it becomes a hexavalent chromium, which is chromium-6, and that is carcinogen. So the idea that I had at that point is, why can't I eliminate this process using a phosphated polyurethane thin film. And so then I went to grad school to do this. And as they say, the rest is history. I was able to design a water-soluble and dispersible polyurethane thin coating for addressing this environmental concern, which otherwise is a carcinogenic material. We have done a lot of studies, as you would expect me. I will not be able to go through most of it, but rheological characterization was very important because the coating applicator here, there are a number of rollers, and these rollers exert a certain amount of shear. And so we have to look at whether the polymer was Newtonian or non-Newtonian or shear thinning or shear thickening, and then we have done quite a lot of studies. So that was basically the beginning of my involvement in translational research, and of course that led to a series of patents, as you can see in here. The first one, actually, we filed it in 1998, as you can see here, and that filing was done in 25 European states or European member countries. In here, if you count them. They are about 25. I believe they are now more than 25, maybe 27 or so. But back in 1998, these were the member states. And then we also went to patent it in 2001 in the US. But then we also wanted to expand the IP protection beyond the US and Europe, and we went to Hong Kong, and there was a reason for doing that. There was a business case, actually. So we protected it in Hong Kong, and then of course in South Africa as well. So this technology was considered to be a very successful one. We have carried out a range of industrial scale tests, and we have documented a lot of data, which were subsequently then licensed and started the commercialization process. So this was my entry into early translational research as a grad student. And I'm mentioning this one just to provide you a context, just in case there are graduate students out there, that it is possible to do some great stuff as a graduate student. And in fact, that is our expectation as well as supervisors. So how did I transition from that into the biomaterials field? Two things came out of what I just mentioned now. The first thing is that I was able to understand polyurethane chemistry and polyurethane processing. That led me to the field of polyurethane biomaterials and scaffolds. And in fact, most of my earlier publications in the area of biomaterials and tissue engineering used polyurethane scaffolds, and that's a reason why I went into that particular polymer. The second important thing that came out of this one is the fact that I was able to use water as a solvent in polyurethane chemistry and the processing of the thin films. Then that led me eventually to work with hydrogels. Of course, as you all know, that hydrogels are materials that are cross-linked, and then they are swollen in water. And so those are the two messages that really came out of that. And in fact, when I moved completely to the biomaterials field, one of the earliest publication that I had from one of my postdocs was this combination of the polyurethane chemistry and the use of water as a medium to design polyurethane hydrogels with improved mechanical properties for biomedical applications, and that was published in this journal back in 2005. Although the work was done 3 years prior to this particular publication. Then the idea when we started working with these particular hydrogels was that this specific combination of butyl carbamate monomer with hydroxyethyl methacrylate would give us a hydrogel that is as good as a cross-linked hydrogel, but it was not chemically cross-linked. It was in fact physically cross-linked. So we went into this area of physically cross-linked polymers, and then we continued to work in this physically cross-linked hydrogel field for a while, maybe for about 7 or 8 years after this. So one of the earliest physically cross-linked hydrogels that we have designed with biomedical application in mind was published in Biomacromolecules, you can see in 2006. And then with this publication, we expanded the idea that physically cross-linked hydrogels can be synthesized quite successfully. So as you all know that linear hydrogels, they tend to dissolve rather than swell. So if you synthesize a hydrogel forming material, and if it's a linear polymer, then it dissolves in water, but then you are not getting a hydrogel material. So that's pretty much useless. And if you then use chemical cross-linking agents, you can transform the linear polymer into a 3-dimensional polymer. But then the chemically cross-linked hydrogels cannot be reshaped. So which means if you want to redissolve it or form a tube, if you want to go into a slab form, then you cannot really do that, because once those cross-links are formed, then they are permanent. One can use heat treatment to see if you can soften the material and you can reshape it. But oftentimes the temperature is high enough that you degrade the hydrogel rather than softening to reshape it. So this option was not an attractive option for us. And so the approach that we have used is to design linear hydrogel biomaterials that cannot be dissolved in water, but that can be processed using organic solvents, meaning that something that swells in water, but if we want to use organic solvent to process it, we can process it. We can shape it, and then if we don't like that particular shape, then we can redissolve it, or we can heat it up a little bit and soften it, because it's physical crosslinks, and those crosslinks can be easily broken, and then you can prepare physically cross-linked hydrogels. So one example here is where we started with benzophenone here, and then 2-propanol. That would give us what is known as tetraphenyl ethanediol inverter, and I'll define what an inverter is here. And this is a photochemical reaction. And then we start with this chemical, which is commonly known as solketal. And then you can combine it with methacrylyl chloride. That would give you solketal methacrylate. And so this is, you can see that the double bonds are introduced. It's a low temperature reaction around 4 degrees Celsius. So it can become a nice base in the presence of triethylamine. And then we, once we synthesized these 2 monomers, then we went into a polyurethane synthesis approach, where we started with a macrodiol, and then a diisocyanate. In this case, it's phenyl diisocyanate. And then we made a polymer which has 2 NCO groups end capped. And again, for those of you who work with polyurethane chemistry, this is not going to be new. And then once we have these NCO end groups, then we brought this inverter. And the word inverter is a combination of 3 words. One is it serves as an initiator, but it also serves as a transfer agent and also as a chain terminator. So it does 3 things. So we brought this one, and through these OH groups, we were able to incorporate these inverter groups into the polyurethane chain. And what we now have is a polymeric initiator, transfer agent, and then terminator at the same time. Then we took the solketal methacrylate monomer in here, and then we were able to create this block polymer. So this bond breaks down, and then it forms a radical, and then it inserts this monomer. And so you can see that you can basically produce n repeat units of this solketal methacrylate in the polyurethane backbone. Now you can hydrolyze that, which is what we did, and then at 25 degrees Celsius and in the presence of reagents, in this case hydrochloric acid, you can actually hydrolyze these groups. That gives you glycerol methacrylate, which is the title of the paper. So this is a polygroup glycerol methacrylate block polyurethane hydrogel material. So this was an interesting study that we have done, and one of my first PhD students was involved in this study. And then we continued to study this one, and in fact we have characterized it extensively. We have done the cross-link density, we have done the elastic behavior, the protein adsorption, the swelling as a function of hydrolysis, and so we have done quite a bit of study on this material. So this was in 2006, and then in 2008, just 2 years after this publication, a medical device company was excited about this physically cross-linked hydrogels, and the company contacted me for a potential hydrogel-based project. At that point, little did I know that there is another world of hydrogels out there. So we start meeting with this company to understand what their intended application for these types of hydrogels was, and then turned it out that it was in radiotherapy quality assurance devices. So as you all know, that in radiotherapy, and you may probably know this stat, approximately 50% of cancer patients at some point will undergo radiation treatment. The standard treatment modalities for cancer are surgery, chemotherapy, and radiation, and no single treatment is sufficient. So at least two of them must be combined. It could be surgery and chemo, or chemo and radiation, or radiation and surgery. So the patient is then placed into this high energy source, a LINAC, a linear accelerator, and then the radiation is released from the machine, and conceptually you're hitting only the tumor, and then therefore killing the cells and shrinking the tumor. So that's what the company was involved with, and then they have a lot of quality assurance devices, and that was the intention, that they wanted to use these hydrogels. So what happens is that as I was learning this process, because as I mentioned earlier, that little did I know that there is a hydrogel field that could be used in this area, I was told that the workflow in a typical radiotherapy process looks something like this. You have a dose prescription that is based on patient anatomy. So this is usually done by a radiation oncologist, and then that goes into then the medical physicist's workflow, where the radiation treatment planning is sorted out, and during this planning there is a dosimeter that is needed, and this is a three-dimensional dosimeter, which preferably is a hydrogel. And so what you're doing here is you have a gel with a radiation tracer molecule embedded in it. And then that gel is then treated as if it's a patient. And that's basically what this box is. So you treat the gel, assuming that it is a patient, and then you do the analysis. And then if it fails, you go back, and then you start the planning all over again. And you continue to do that until it passes the prescribed radiation delivery procedure or prescribed process. And then if it passes, then of course you take it to the patient treatment. So that's basically how the workflow looks like. And then this is called end-to-end quality assurance, because you're going through this loop until you actually get the exact, or at least as close to the ideal treatment plan as possible. So we have been working in this area. So what is a problem in here? Well, clearly prescribing the dose is not a problem. Delivering the dose is not a problem either. We have machines that can deliver that. But the problem is, how do you assure the patient that the prescribed dose and the delivered dose by the machine actually ended up to the target where it's needed? So the goal here is to design a dosimeter that can predict that if you deliver it in such and such way, that you can actually reach the maximum at the tumor site, therefore sparing the vital and healthy tissue surrounding the tumor. So that's the idea. So we've been working in this field for many years now, and these are some of the publications that we have done over the years. And then the whole idea in here is to just to design that gel forming material. So what material to pick, what tracer to use, and so on and so forth. So the design criteria for these gel dosimeters is that it has to be a soft tissue equivalent hydrogel, because if you take tumor, the vast majority of it, over 95% or so, is water. So your hydrogel must be largely made of water or contain water. And you can't have heavy metals in there, because in tumor, again, you have nitrogen and oxygen and hydrogen and carbon. So you don't actually get heavy metal. So you cannot really use a material that has a high electron density. So that's one design. So the hydrogel must be, the material must form hydrogel at low concentration. Then we have to embed a radiation tracer compound, because that's the one that is going to be sensitive. And then, of course, there are additional criteria here. It has to be optically clear. And the reason for that is because the imaging is done using optical scanners that's the preferred way of doing it rather than MRI, because MRI is very expensive. It takes patient treatment time. And then, therefore, it's useful if we can do it using optical scanners. And then, therefore, optical clarity was the design criteria. It has to be sensitive in the clinical dose range, and that goes to the radiation tracer molecule, because the molecule has to be sensitive to radiation from 1 to 80 gray, and gray is a radiation unit here that is joules per kilogram. And so that's one design criteria. The reason is that you get a lot of radiation sensitive molecules, but they're not sensitive in this low dose range. So it's really important that they are sensitive. And also it has to be stable. Once the image is formed, then there has to be, the idea is to have zero image diffusion, and I'll explain it to you by what that implies. And of course, that's to have a fast response, easy handling, and et cetera, et cetera. So if this is a tissue, if you just were to imagine that this is a tumor in here, and then you have ionizing radiation coming from the delivery, and in this case, the radiation is coming from three angles, 1, 2, and then here. And so the maximum is going to be at the center here. And if there is a radiation sensor in this hydrogel embedded, then you will find these images formed, because the radiation sensor molecule will absorb the energy and then it changes color, and then you can image the color. By just looking at the position of this color, you can tell where the delivered dose is actually located or maximum in this tissue space. And that's really the idea behind quality assurance. Now, of course, the radiolysis of water is the main reaction there, because tumor shrinkage or killing of tumor using radiation involves oxidative damage. So the high energy radiolysis of water forms all sorts of radicals. They're very chemically reactive. Some of the chemically reactive radicals themselves recombine to produce a more potent reactive combination, such as superoxides and peroxides, and ultimately that produces an oxidative damage to cells, and then that's how you kill cells. So these are basically the design criteria that we have, and moving forward, we started with two different gel forming materials. The first one is iron gelatin combination, and so we used the gelatin less than 10%. And then we have other additives in here. Xylanol orange was added just as a color forming compound to form a complex. When iron 2 is converted into iron 3, that's where the image is actually formed. And then we have used cross linkers. This field has been going for quite some time. our contribution to this area is that we were the first one to propose to use glyoxal as a modifier for the gel. Now, initially we used it for a different purpose, but it turned out to be actually quite useful in terms of optimizing this particular hydrogel. Glyoxal, as you may know, is the smallest dialdehyde, and then we use that one, and so we combine this gelatin with either xylanol orange, or we also used methyl thymol blue, which again is a ligand that we have used it in our lab for the first time, and you can see the footnote in here. And we basically went through a whole lot of optimization in terms of the lambda max. So, for example, if you don't add xylanol orange and you take iron 2, and in the presence of oxygen, you form iron 3, that's basically the oxidation process, then the lambda max is 310, which obviously is not useful. But then if you add xylanol orange or methyl thymol blue, you can shift the lambda max to 450 or to 585. And in the case of methyl thymol blue, you can actually do it at 620. Now, the reason for this is because in optical scanning, the lasers that you are using, generally it's either a red or a green, and 585 is not convenient. It's somewhere in the region of like orange to yellow laser, and that's not really very common. At least we haven't seen those things. And so there is a better signal detection at 620, and although there are other techniques such as LEDs that can be used as well. So we went through this optimization process, and we were able to demonstrate that, in fact, some of the design criteria were met, particularly the fact that it was tissue equivalent. It's because it's gelatin, and it was very sensitive, which was exciting. But one of the challenges that we have is signal diffusion or low chemical stability. Now, if I just take you back, that the diffusion, what it means is this is what you will be imaging after you irradiate the sample. When you image it, if this molecule stays where it is, then you know the radiation path. If this molecule starts then diffusing into the other tissue space here, then obviously it's at a different location now compared to where it was when it was first hit by the radiation. And that's really how important it is when you talk about zero image diffusion, meaning that once this color is formed in the hydrogel matrix, then it has to be frozen there. It shouldn't really move, because if it moves around, then it's going to give the wrong information. And then when you deliver it, you're not actually hitting the tumor, you are actually hitting potentially the healthy tissue. And that's how important this design criteria was. And so we were partly successful in gelatin, but then we decided we could do more exciting stuff. And so we went back into designing different sensors, because the gelatin was a soft gel, and the iron molecule, although we have done the best we could, we were not able to stop the diffusion, and the stability was also a little bit challenging. And so we went back to the drawing board, and Kellen Penev, who was a postdoc at that time, and he is a research associate in my lab now, he went into tetrazolium chemistry and gel and gum gel dosimeter. So now the gel forming material is now this naturally occurring gel and gum, which has a tetra 4 sugar molecules as a repeat unit. That's the typical repeat unit for gel and gum. You have 2 glucoses, this one and this one, and then you have a few other sugar molecule, and that is a repeat unit. So it's a naturally occurring material. It's done. It's produced by a fermentation process. So we chose that gel because it forms a stable gel at low concentration, and then we have synthesized through a very lengthy chemistry. And if you are interested, I invite you to read this paper, which is Journal of Heterocyclic Chemistry that we published it in 2015. It's a multi-step process to actually get this radiation tracer molecule that can be activated using the clinical range doses. And then we have added a few radical scavengers and some cross-linkers, which is a lithium salt in this case. And we were able to meet most of the criteria that we set at the beginning using this particular hydrogel system. And again, the details are published in these papers, and so I invite you to go and have a look at it if you are interested. So what came out of that is some years back, we were able to transfer this technology to the company Modus Medical Devices. They recently changed it, their name to Modus QA, and you can see the website down here. And we actually licensed two different hydrogel systems with two different tracer molecules. One was a gelatin-based cross-linked with glyoxal, and another one was gel and gum, and the radiation tracer molecule is a tetrazoline molecule. And so they have been doing their own pilot trials. We assisted them to scale up the synthesis or the preparation of the hydrogels and the cross-linking and all that. And they have done their own industrial tests, and recently they launched this product as a ISO 13485 compliant product. And now they have started putting specifications and some of this information that you can see from their website. We actually extracted that. This is basically their product specification that is required for getting this ISO certification or the ISO compliant information. They also have done real clinical studies using that one, one of the gels. This is the tetrazolium one. One is in the planning of cranial stereotactic radiosurgery, which is shown in here. But they also have tested it using intraprostatic ablation here. And then they published their own findings down here. But they are quite happy about the technology, and we are expecting that this technology will be widely utilized in the near future. Currently, we are optimizing this research because there are still room for optimization, and we are testing new tetrazolium salts. And you can see that this is the molecule that we have synthesized and we have used, but we can also prepare different sensor molecules, and the gel forming material is still gel and gum, because we found that gel and gum is actually the most user-friendly gel, just simply because it forms a gel at a very low concentration. Something like around 1.5% would actually give you a stable gel. There are also other requirements, as you probably expect. Doing science in the lab is sometimes easier than translating or meeting some of the industry criteria. So one of the criteria that we were told at the beginning was that the gel should not really melt or soften if the temperature increases from, let's say, 25 degrees Celsius to about 50 degrees Celsius. So my natural reaction is, why do we have to worry about 50 degrees Celsius? And it turned out that if this material is shipped into some countries outside Canada where the temperature in that particular country is very high during the summer months, and the warehouse is not properly ventilated, the temperature could reach that high, and then therefore, if the gel softens, then the product will be difficult to use. And there are reasons for that. I don't want to get into that one just in the interest of time. But these are some of the criteria that we had to work with, and then we still feel that we need to optimize it. So we are exploring different chemistries in here. Some of them are published, and some of them are still in the process, the ongoing projects that we have. So overall, we believe that it's a quite successful project. So because I was working with, or just my lab generally has been working with gelatin, we have been working with collagen and elastin fibers and elastin gels from previous studies that we did. My lab also has been working on conducting materials for scaffold fabrication and tissue engineering applications. Around the time where we licensed the gelatin hydrogel, and we have now that experience, I took a sabbatical, and that's actually the best sabbatical that one could ask for. And then I started collaborating with Professor Malcolm Shing from the University of Manitoba. We have known each other prior to that through the Canadian Biomaterials Society and then other conferences. And I know that he was working with gelatin and gelma and collagen and elastin and conductive materials and egg white gel. So he was working in all these areas. I was working in the areas that are shown here by red. And so there was a common interest to explore these materials. And we started collaborating with Professor Shing, and it produced, some exciting studies, some of which are listed in here. So the idea is where can we use some of these materials in a different way. So for example, my lab has been working with convincing the smooth muscle cells to produce elastin. I was incorporating reduced graphene oxide into scaffolders so that we can differentiate stem cells into particularly cardiac stem cells into cardiomyocytes, or neural precursor cells into neural cells. So obviously those tissues, they do require some electrical signals, so we thought that we could use conductive materials. So I was working on that one, and so was he, but he was working with other materials as well. So because of this complementary expertise that we have, we started working, as I said, together. And what I'm going to do is just give you a couple of examples of what came out of that study that is quite exciting, and that we are in the advanced stages of licensing as well. So one thing that we thought was, can we design hemostatic adhesive gels? We know that tissue adhesives are very important. They could potentially eliminate sutures and staples following surgery. But at the same time, they could also have therapeutic benefits in a sense that these gels, they not only seal a post-surgery tissue, but also they could potentially be life-saving in a situation where there is excessive bleeding. So to do this research, we were motivated by a few things. Number one, in battlefields, as many as 25% combat mortality are potentially survivable before arriving at a hospital. If, and that's a big if, there is a material that can be applied that is easy to use and that is very quick acting in stopping bleeding. So that was the first thing. As you know, that in combat situations, the soldiers are oftentimes far from a hospital, and so until they get to hospital, most of them, they actually die from bleeding. They don't die from infection within that short period of time, but bleeding is the main cause. Number two, every year, over 26,000 civilians around the world are injured by fragments of improvised explosive devices, and most of them die from it. In addition, other incidents, for example, severe bleeding that can lead to significant blood loss and potentially death, they include blunt and penetration, trauma such as accidents, car accident being one example, and even injuries from hiking and climbing tools. So while these injuries are real, available products to stop bleeding and save lives are either not effective or are complex to be used by first responders, battlefield combatants, and by ordinary people in household setting. So the idea is, how do we develop a material that is easy to apply, that doesn't require excessive tools or accessories, but that can stop bleeding. So based on prior knowledge in the field, we have identified some materials. This is gelatin and functionalized to form gelatin methacrylate. And then we have used triternalamine, vinyl caprolactone, and then eosin Y, which is a visible light activated cross-linking or initiator for cross-linking. And we took then an enzyme from a venomous snake, and this enzyme is repitalasin here, basically an enzyme that is extracted from the snake. And then we embedded that one where we loaded that, and that paper came out about 6 months ago, in this title. It's published in Science Advances. Of course, then we characterize the hydrogens just to make sure that the loading of the particular enzyme is not changing the morphology, and we have done, as you would expect, the standard characterizations, and just looking at whether this material is good to stop clotting. So the first thing that we have tested was the adhesion strengths, because for any tissue adhesive to function, it's important that it actually sticks to the tissue. And so this is an in vitro test where we took a porcine skin, and it's an end-to-end adhesion strength test. Basically, attach two pieces of porcine skin using this glue, and then you pull it apart after a given period of time, and then you measure the strengths in terms of pressure force here. Fibrin glue, we used it as a control because that's the gold standard that's clinically used now as a surgical glue. Gelma, because it's been reported in the literature to have some excellent adhesion strengths. And then this is the hemostatic adhesive, which is HAV, our formulation. So you can see that the adhesive strengths of this new gel was as good as Gelma, which has been reported to have, as I said, excellent adhesion properties, but it was much, much higher than fibrin glue. So we were excited about that. And then we continue to look at the blood clotting assays. And again, a lot of data. I apologize. This slide is crowded, but the message is very simple. This is an in vitro blood clotting. So we in a culture well. So this is a control where nothing is added to the blood. You just pipette blood into these wells, and then you look at as a function of time when they form clots. And then as you go down here, you can see that you have gelma here, you have the hemostatic adhesive, which is our material, and then this is a fibrin glue. And so you can see that as early as 30 seconds in here, that's what you see in here, that the clot was actually formed. And then when we look at the clotting time, this is a control. It took about 5.5 minutes before a clot was formed in the control. That's nothing added. And this is just the repitulase alone, which is the enzyme that is extracted from snake venom. This is gelma, and this is our material, and this is fibrin glue. So you can see that we were able to reduce the clotting time down to about 45 seconds. If you look at this axis, it's about 45 seconds. So in 45 seconds, blood is turned into a clot, and then therefore it seals the surrounding tissue. That's the idea. And you can see from the SEM images, it's our red blood cells, as you can see by this shape images that you see. Down here, what you will see is as a fibrous structure, and that's of course in clot formation. We know that fibrinogen is converted into fibrin mesh, and that's basically for part of the clot. Then we took this in vitro data, and then we did a blood loss and a clotting assay, and you can see that we were able to demonstrate in a rat tail amputation model that the amount of blood that is lost was minimal for the hemostatic adhesive that we have developed, as opposed to the control or the enzyme alone without the gelatin methacrylate matrix. And it was also much lower compared to the fibrin glue, which is of course what we want. Now, in terms of time, and again, the timing was actually the shortest for this one in a rat tail amputation model. Then we went and we tried it with liver. The message was the same. You can see just the red bar in here is actually the loss, and that's the amount of blood that is lost after the incision was made. And this is the time that it would take for the blood to form a clot. And then again, you can see that this is very, very short time. And practically what it actually means is that if someone is losing blood and you apply this gel, you can actually stop the bleeding within such a short period of time, and then you lose very little amount of blood in the process. So not only we tried it with liver, we also tried it with arteries, and we basically ruptured artery, and then blood is coming out, and then we apply the sealing, and we were able to demonstrate that this material is actually quite an exciting material. So when this paper was published back in July, we were excited. It picked really a lot of momentum, and you can see that some of the scores that have been compiled by the journal, that it was in the top 5% of all research outputs scored by altmetric, and it made news not only here in Canada, but in the US and in Europe, and even in other parts of the world, which was quite exciting. Now, one of the issues that we have was the hemostatic adhesive gel that we have developed. First of all, it's actually cross-linked by visible light. I forgot to mention that. That was actually one of the benefits as opposed to a UV light, because UV lights are not always safe. Because it's a UV light one, one would imagine squeezing this gel, which is a liquid-like material, and then you shine a visible light, and then that gets activated because of eosin Y, and then the cross-linking process starts, and that's basically how it stops the bleeding process by forming gel. Now, that depends on how quickly one shines the light on the gel following, the squeezing process onto the site of the injury. And, sometimes if you look at from a practical standpoint, one of the things that you would like to see is would a person who is bleeding be able to pick up that adhesive or that glue from a tiny container, squeeze it, and then shine a light within a very short period of time? Because if that process is not fast, then it gets washed out, because there's body fluid, there's blood all over the place, and so washout becomes an issue if the person doesn't actually shine the light very quickly. This is actually a condition where the gel itself is solubilized before it forms a hydrogel by the surrounding fluid, and then it doesn't really stick onto the tissue. The second issue that we have observed was potential of swelling, because it's gel, it swells, and when it swells, sometimes it gets heavy, and depending on the anatomical position where you apply it, it could exert enough weight that it could detach as a result of the swelling process. And the detachment it could be life-threatening, especially in vital organs such as the lungs, the liver, and the heart, because these organs generally undergo frequent deformation or movement due to physiological needs. And so it's not a static tissue, so it's really important that that is also avoided. So in a follow-up study, what we have done is complementing the repeatedless hemostatic adhesive gel, in a paper that came out also in Science Advances a couple of weeks ago, where we have designed a hydrophobic, which is water-hating, to address the swelling issue and this washout, because if it's not solubilized, then it cannot be washed out. A water-hating, highly hydrophobic in situ hydrogel for tissue adhesive. So this material functions in a very interesting way. Basically gets into the site of the injury, displaces water at the site of the injury, makes contact with the tissue, and then it forms a strong adhesion. And then therefore there is no issue of detachment, swelling, or washout. So we were able to address these issues that we observed in the previous study in this follow-up study. And then these are just some of the addition strengths. So you can see that you can take this underwater in situ hydrophobic adhesive, and then you can put it, once the addition is formed, you can put it in water for 24 hours, and then you measure the adhesive strengths, and you can see that, in fact, it improved compared to the one before. Fibrin glue, you can see that the addition strength is actually low to begin with, and then after 24 hours, there is no improvement. So it's another exciting study on this one. I'm just going to show you a couple of videos just on this one. Well, it doesn't seem to work. If you go into the journal, you'll see the first one is a self-healing one, and the second one is a video. Oh, that's unfortunate. You'd actually see that a balloon that was filled in this particular animation, a balloon that was filled in water or with water, and then and you'll see that there was a puncture that's made, and then this adhesive was applied, and then you'll see immediately that the balloon was no longer leaking. So it was an exciting one. Yeah. I'm sorry that I couldn't show you the video for some reason; it's not working. So in terms of translational pipeline, IP protection is underway for this particular technology. We are, as I mentioned earlier, in an advanced discussion with biopharma and different biotech companies. A number of companies expressed interest, and hopefully we'll see this one translated hopefully in the near future. Now, let me just circle back because I mentioned two things, and I have only a couple of slides, and then I'll come to the end of my talk. I told at the beginning, I mentioned at the beginning that transitioning to the biomaterials field were contributed by these two factors. So the first one was understanding the polyurethane chemistry and the processing, and the second one was the hydrogel. So far what I talked about is all to do with hydrogels in one way or another. What we have done was these polyurethanes over the years is actually quite interesting. We have developed, and this is really where I get into the vascular tissue engineering area that I said that I will not be talking, and I only have a couple of slides in this one. So where we have fabricated scaffolders, we seeded vascular cells or multipotent progenitor mesenchymal cells, and we put it into a bioreactor. We mature it for X period of time, and we have been publishing since 2007 in this field, and we really have found some interesting things, and the polyurethane biomaterials actually served as an excellent model biomaterial. Polyurethanes can be designed to be biodegradable, but the ones that we have used is biostable, because when we did our signaling studies, we don't want any degradation products to confound our data interpretation. So one of the challenges in vascular tissue engineering was the absence of elastin production, and we were able to use different techniques to produce elastin, and just to compare it with rat elastic tissue here. The top one is a control, and the bottom one is our product, and you can see that in the engineered tissue, elastin was expressed quite abundantly. We also looked at elastin cross-linking. When elastin is cross-linked, it requires the scaffold protein, which is called fibrillin 1. And every time we do these tissue fabrications, where we see elastin, there was also fibrillin, and then therefore this indicates that, in fact, we were producing a vascular tissue which contains cross-linked elastin. And so we have been doing other things as well. We have done signaling analysis or signaling pathway investigations as to why elastin is produced on these polyurethane surfaces, and we compared 2D film, which is a very thin polyurethane film, and this is a 3D polyurethane scaffold, and you can see that the signaling pathways that we observed just by changing the topography was just completely different. Every time you see one arrow going up, it is down for the 3D and vice versa. And so we were able to demonstrate that elastin biosynthesis in an engineered vascular tissue followed these pathways, and that was published some years ago. I think in the interest of time, I'm going to stop in here. Recently, we were also looking at developing a technology in injectable and conductive cardiac patch repair for myocardium repairs. And I think that's just looking at the time. I think we'll probably stop in here. So with that, I would like to acknowledge the funding sources, as you can see, and the work that I have presented today. Most of it was the hydrogel stuff. The early hydrogel work was done by Alpesh Patel, who was one of my earliest PhD students, and now he is a senior scientist in one of the biotech companies in the US. And so he did the physically cross-linked hydrogel stuff, and then the early dosimeter work as well. Then the rest of the dosimeter work was done by Kellen Penoff, and this is him that you see. And I think most of the studies that I showed you today in terms of translation are contributed by Alpesh and Kellen. The last in production and the signaling work was conducted by Shigang. He's a research associate in my lab. He is still in my lab, and we're happy to have him. And of course, other students contributed in one way or another. So with that, I would like to stop in here, and I'll be happy to take up any questions that you may have. Thank you.
Many thanks, Professor Mequanint, for this inspiring talk. We learned a lot from your research, the history, and I really liked, the picture that you showed us in the beginning. So before we go to the questions, you can stop sharing so I can just click on that cross in the middle on top. Yeah.
So I'm trying to find where it is. Okay, so I just need to go to.
Exactly. So before going to the questions, so I just advertise our next speaker, and then we can start. Oh, so I think you have another page open, like. Okay, so let me just see. Okay, so I can't share, so I go to questions from the audience. Thank you so much for joining us, the audience. I think you explained about the design criteria for the 3D dosimeter, but I think also you explained about the zero image diffusion. But one of the audience is, John is asking why zero image diffusion is the criteria for that.
Right. So obviously, zero image diffusion is not possible. So that's more of like a desired criteria because diffusion by its very nature cannot be zero, right? It can go to very small numbers. So why is it important? The reason why it is important is you have a tumor, and you basically would like to use this dosimeter as a surrogate for the tumor, and you want to deliver the radiation through certain angles into this gel. So as the radiation is hitting the gel, the radiation sensor that is embedded in the gel will be turning into a different color, and that color is what you are imaging. So you know where the radiation was actually, maximum in a tumor born. And so let's say that you have one radiation sensor at location X when it was hit by radiation, by this high energy ionizing radiation. And then it changes color at location X. But then you have to take that thing out of the radiation unit and you have to image it. So between you irradiating the sample and you imaging the sample, if that molecule diffused now, it's basically showing it as an image at a different location. And since your planning and your interpretation is based on where that image is at that point, which is the point where you are imaging it, then you are basically getting the wrong information. And that's the reason why it is important. So as soon as you hit it with ionizing radiation and it changes color, if it stays there, then you know exactly where it was at the beginning. So that's how important it is. So if you just moved into location Y, then you go and you image it and you basically say that, okay, well, my image is developing in this portion, and then therefore I'm going to plan it this way, then it could potentially be wrong. And then that's really the issue. So zero image diffusion is more of like a goal, an aspiration rather than a possibility, because molecular diffusion cannot be stopped, as we all know, right? But that's the idea. There is a limit, how much you can diffuse, but we call it, zero image because it's kind of an attractive term people would like to hear.
Thank you so much. I have a question about the same gel, dosimeter gel. So you showed us that the company pursued this kind of technology to clinical application. So I can imagine that they inject this gel inside the animal or human, right? Is that right?
They don't. So basically, they don't. They actually use it to plan the treatment, and that's why it can be used without, like an extensive approval
process. So you don't need like a strict sterilization process for that.
No, you don't. So if I just use like something, this is hand sanitizer. Okay, let's say that this is my job, right? And so this hydrogel has radiation sensor molecules embedded in it. So I am now basically treating that this is actually the tumor. Okay, now obviously people use other things. I didn't get into the physics of it, how this gel can be positioned into a plastic man, for example. If it's a prostate, it will be in a prostate location, and you basically slide that, and then now you're irradiating it, this gel. And then you take it out, and then you image that gel, and you know exactly where the maximum radiation is deposited in that tumor, and says that, okay, if I deliver it in such and such way to this patient, this is what I'm going to get. And so you basically plan your treatment, and once you get a good treatment plan, then you forget this guy, and then you go to the patient.
Perfect. And these gels are acellulars. You don't have any cells.
They don't. Yeah. Okay. You just have to match them with the electronic density, meaning that it should have a little bit nitrogen because there is protein in the tumor, and a little bit of a lot of carbons, and then of course a lot of hydrogen.
Okay. So the next question from our audience, Dalia, is asking. First of all, she's thanking you for your great talk. She wanted to know about the biodegradation rate of these bio-adhesive gels.
We have done an in vivo biodegradation. This is I'm assuming that this is to do with the bioadhesive one, right?
Yes, exactly.
Yeah, we have done it over a period of 28 days, I believe. The hydrophobic one, the follow-up study that addresses swelling, detachment, and washout, that is stable because the hydrophobic starting materials are silicon-based materials. These are actually PDMS-based and the silicon macromolecular fluid. So they are stable, at least for the times that we have investigated. The Gelma one, obviously it degrades because it's gelatin metacolate. Yes, it's a cross-linked hydrogel, but it does degrade a little bit over a period of time. And as long as it's biocompatible, unfortunately I didn't show those videos, but if you would see that the amount that we put is small enough, and then you just basically close, the wound, and then the material stays there. So if it degrades into gelatin fragments and a few things. So gelatin metacolate is one of those materials that have been studied by many researchers, and we know that it degrades. It's just a matter of time. But in terms of hemostatic properties, the important thing is actually the first couple of hours until the person gets into a hospital for further treatment, or until the clot is formed. So, for example, if there is a chef in a high traffic kitchen, for example, and then then there is an accident and he cut his finger and the cut is deep, you just apply that, and until the after the fibrin mesh is formed, then the clot is sealed, the bleeding is sealed, right? So in terms of degradation, the healing process will take over shortly after blood is stopped. So the goal is to stop bleeding here.
And I can imagine that these gels are just for external usage, like wound healing and this kind. If we go inside, for example, for defects in the heart wall or these kind of things when we have the turbulency of the blood and the stuff. Can we apply such a thing there?
Yes, yes, it can. I have a number of videos, but I would encourage the person asking this question, if it's not you.
No, it was mine.
Okay, that's good to know that. There are a lot of videos uploaded in as supporting information to these publications, and we have exposed, for example, an artery, a thoracic artery in rats, and you would see that. Actually, it was in one of my slides, by the way. I just moved a bit fast, and then you basically make an incision, and then just blood is just coming out at that pressure, as you'd imagine. And then you just apply that thin layer, and then you cross-link it, and it stops. It's actually a wonderful material. Yeah.
Perfect.
And we did liver as well. We did a portion of a liver, cut. This is just a mild injury model, but we also have done an extensive liver injury model where the blood is flowing at a much higher rate. And then you apply the gel, and again, it stops it within a short period of time. I was not exactly sure if the audience can withstand, that level of blood, like on your liver, so I didn't show it, but it is in the paper.
Okay, I'll go and check it out for sure. And there is a question about, I don't know, Nasir is asking, could you briefly explain why washing out was a problem for the gelatin gel?
Okay.
I'm not sure. Yeah.
Yeah, so it was not a problem when we applied it, but it is a problem if we want to translate it, because it's all about a matter of time, right? So suppose that this is the gel. I'm just going to use this one, if you don't mind, just to demonstrate.
Yeah, of course.
I like using some tools. So suppose that this is a gel. So now I'm basically squeezing this thing, and it's just going to come out. So it's a very thin layer. This is where the bleeding is. And now I'm going to take a visible light, and I'm going to shine that light, and that activates the gel, and it forms the gel formation, right? But before the light is applied, it's still liquid, right? So how quickly you apply that light, it really matters, right? Because if you just wait, trying to find, you apply it in here, and you're trying to go into a drawer or something, trying to find, where the light is, by that time, this liquid-like gel-forming gelma is probably washed out, because there is body fluid, there is blood, and then that kind of thing. So if you apply it quickly, it's not a problem. So of course, for this paper, we applied it so quickly, very, very quickly. And then again, there are several videos there. You would see that we didn't have a problem. But practically, if I use this one, for example, as a first aid kit in my kitchen, right? And then there is a cut. Even just to get like my cell phone flashlight on, it does take some time, right? And then there's nervousness and concern and all that. And so during that period, it could get washed. So we thought that if when we translate this thing, this becomes an important consideration, because it boils down to time and how effectively people can use it. And that is really what we're trying to address in the follow-up study, is how do we eliminate this washout? So by using this hydrophobic material, now you can eliminate that, because there is no issue with, shining a light. It basically forms hydrogel by itself. It's silicon based. It's hydrophobic. It doesn't swell. It doesn't detach, and it doesn't get washed out, because it's no water soluble. So that's the idea behind it. It was not a problem for our study, though.
Of course. Thank you so much. There is one question. I don't know if it's pertinent, but Ali is asking, first of all, thanking for your great presentation. Have you tried using this, using organelle chips for studying the performance of these hemostatic adhesive gels?
No, we did not. It's a good question, and we haven't actually used that one. We did quite a number of animal models, and just to add one point, the hydrophobic material, for example, we did it in 4 different animal models. We did it in rats, we did it in rabbits, we did it in dogs, and then we did it in pigs, in different things. So we used it to fix to seal a fractured skull to simulate bone. We used arteries. We used the skin, and some of the videos that I didn't show were actually showing that. So we did multiple tissues, and we're pretty happy in terms of the mechanism and the way it functions. But as an additional study, organ on chip would be a useful thing to look into that. But we can only look into that.
Perfect. So I have a question myself, because you briefly showed us a study in Nature, I think Biomedical Engineering, that or materials that cardiac patches, right?
Yeah.
So, and using, I think, reduced graphene oxide as a conductive material.
It was carbon nanotube, yeah.
Oh, carbon nanotube. So
Mm-hmm.
there's always a concern from especially the reviewers of the grants about these using these nanoparticles and the circulation in downstream or accumulation in the heart or in downstream tissues. How do you answer that? So then I can learn.
Yeah, that Nature BME paper took forever, just partly because of the reasons we had to do actually 6 months in vivo studies.
Yeah.
To look at the distribution of the fate of the nanotube. Let's put it that way.
Okay.
Basically, the material has elastin. It's got collagen. It's got carbon nanotube. You can squeeze it. It's injectable, and once it's injected, then it recovers from the deformation because it has to recover from that one. So it's a shock memory material, and once you adhere it to the cardiac muscle, then it has to be conductive, which it was. So conductivity was not interrupted by that. So a lot of great stuff. But then what happens to carbon nanotubes, right? Are they going to stay there? So for the large part, they were confined within the environment in which we have applied. And then we did 6 months in vivo assays, and we look at the lung, the liver, and the spleen, and some of these vital organs. And then there was no evidence that there is an accumulation in one of these organs. So that explains why it took so long, because when you have to do like 6 months of in vivo studies to show that, it does take time. But it's an excellent question.
Yeah, exactly. And another thing is that how the integration happens. Where you have this fibrotic tissue after MI, and then you inject it, and then so you have kind of barrier, and this integration of the hydrogel to the wall is in terms of conductivity, electrical conductivity, and all this integration. It happens around the border, or
it happens on the surface, right? Because
surface,
you're applying it on the fibrotic tissue, so you have the healthy tissue from the left and the right, and so as long as the conduction wave is able to transmit over that material, then we were able to show that. We have done the performance of the ventricle after that in terms of fractional ejection fractions, and, the thickening of the wall, and all sorts of things. And so that is that's well studied aspect of it. And then we did also the electrical integration. One of the things that I should mention in that study is that over a period of time, there was still some evidence of fibrosis, particularly for the long-term implants that we did. And that still requires optimization. I don't want to deceive anybody here by saying that it's perfect for all time points. It was perfect at the beginning, and then there is some evidence that it is weakening. The conduction was still there, but it was weakening because of the fibrotic tissue surrounding that.
And did you have the vascularization through
yes.
the?
We did the vascularization. I don't think that we have done vascularization specifically within the patch itself.
No, from the heart after transplantation over time, from the heart, like that the heart start, angiogenesis or vasculogenesis.
We looked at recruitment of resident cells into the injured site. Yeah, we have done that. Yeah.
Perfect. These are really cool study, and it's my interest also to look at the paper. I think I look at the paper, but I have to go in details.
Yeah.
So thank you so much. Yeah.
There is 50 pages of supporting information. I think that's why
Yeah.
a lot of these things there.
And you didn't have cells inside the gels.
No, we have. In some of them we have. In
So
some of them we have.
do you need immunosuppressant kind of material, kind of medication for the animal?
No, we did not. So we had rat cells, and we have also other cells. Some of the gels, the conductive material, the patch itself has cells in them. Some of them are acellular, meaning that there are no cells. Some of them have elastin collagen. There are about maybe close to 7 or 8 different materials, or maybe even higher. We basically looking at the carbon nanotube loading, the ratio of elastin to collagen, because that was really important in terms of shape recovery following injection, and also the injectability. And then some of them we have cells in them, and then we did cell viability after injection. Because basically this is not a suspension, right? In a hydrogel, in a typical injectable material, it's not liquid. It's basically a preformed device that you're collapsing it into a catheter or into a syringe, and you're basically pushing it through. So we looked at cell viability and things like that there as well.
So Kibret, we got some questions from Professor Blaschak. He was one of our previous speakers, and he's working on vascularization angiogenesis, and he has a small molecule and in a company. He's saying that come back and present the cardiac work another time.
I would love to. It is we had a lot of interest as well, and for that work as well. We are in the early stage of the IP protection, and if there is a platform to present it, I mean that's basically a seminar by itself. It's a very large.
So
study.
I can ask you if you have time, maybe in the fall, come back for this presentation of this cardiac work, because it's also my own interest and also audience. And also our own colleague, Jianyu Lee from McGill, is thanking you for your presentation. And of course, we will have him also as a presenter in this seminar in the future. Thank you so much. And this is our time today. And thank you so much for your presentation, and hope to catch up soon, maybe in fall, for kind of progress in cardiac work or other translational work that you're doing. And have a great day. And thanks all the audience for
inviting me. Yeah. Thank you.
Thank you. See you. Bye-bye.